Good afternoon, everybody. Welcome to Life Sciences Grade 11. My name is Duduzile Kupeka. My email address is dkupeka2204@gmail.com. The topic is still on the human urinary system. So yesterday I promised you that I'll show you a video where they are dissecting a kidney. So first up, I'll just show you the experiment as it's posted on the Via Africa Life Sciences book. This is basically how you should follow the instructions when you're going to do it. Then I'll play you a video, of course, where they are it's a crazy country and it's crazy times, but guys, it will pass. Please just focus on your work. Avoid all distractions. I know there's a lot of confusion going around, but keep your focus. That's the most important thing. The end goal, you know what you want, you know what you're going for. So close yourself off to all the confusion happening around you and do your best to control what you can control. You have access to online material, you have access to textbooks, you have access to everything you need. If you don't have access to certain things, ask for help wherever you can ask for help. But just do your best to make sure you at least are ready for your exam and pass. And then that topic here on the kidney. What are the parts of a mammalian kidney? I've already described them to you and we've discussed this together in class. But today we will see a kidney being dissected. So the aim of this practical is to what? To determine the external and internal structure of a mammalian kidney. What you need, information sources, fresh sheep kidney, a sharp knife or scalpel, petri dish or saucer, hand lens, glass rod. The information page following. So we'll see the information page, right? What you need to prepare by reading and viewing the information page. Note the external shape, color, and size of the kidney. You'll remove any fat, slice the kidney across the middle as shown, use the hand lens to look closely at the kidney, then draw a neat diagram of what you see. Twice actual size of the kidney, identify and label the parts. Push a blunt glass rod into the urethra and identify the position of the pelvis. You will draw and annotate a hypothetical diagram of a cross-section of a mammalian kidney using your findings from the longitudinal section. Then you will prepare a scientific report on your investigation. I covered that part last when we did the other topic. I showed you how to do your aim, hypothesis, your materials and methods. Then you do your discussion. You go forth to having your conclusion, and then you reference your work. So please remember that. And it's all important. It might not seem relevant to you right now, but if you want to be in, maybe you want to study a course in the sciences, even at varsity, you will definitely be doing a lot of scientific reporting. You'll do a lot of experiments and you'll need to write up reports every freaking week. So get used to it. And if you really want a career in this field, I'd imagine that excites you. So just look at how you feel about certain things. That will also help you direct you in certain careers. If you see that this is basically what you'll be doing in your career, then maybe it's not something you should go for, So monitor that as well, not just how well you are doing in a subject, but also monitor how you feel when you have to do certain things that would be required for you to do in your career. That will save you a lot of time wasted. So yes, take your career guidance very seriously. It's important. It will determine the rest of your life. And then now, the ethics and hygiene. There are ethics with everything. There's ethics we need to adhere to. And particularly because now the kidney is obviously an organ found in living organisms. So it means there are some ethical issues we need to take into account. Sorry, there are some ethical issues you need to take into account. I'm very sorry about that sound. Okay, sorted now. There are some ethical issues you need to take into account when you're going to deal, especially with living organisms, because you can't just do as you please. These are lives that matter, and you can't just do as you please with them. So you'll need to remember that every dissection must consider the ethics of scientific research and use of animals. So get familiar with those ethics. You can't just kill for the sake of killing. You can't really just kill a large, imagine you'll just kill a kidney simply and a whole sheep just because you want the kidney for an experiment. No, it doesn't work like that. Dissections should be shared by many people. Do not abuse living organisms unnecessarily. Dissections are for scientific research purposes. So always remember to consider hygiene factors too. Ne? So what are ways you can maybe get your kidney? Like I said, it wouldn't be, imagine you can't go buy a whole sheep just for dissecting. So what can you do? Maybe because you know there's a butchery around you, they are obviously selling that meat. They are selling lots and lots of meat there. So you can go there and buy maybe the kidneys that are left over, or maybe they'll even throw them out. Others sell them, others throw them out. So you can consider that option and not kill another sheep unnecessarily so. You understand what I mean by that? But it's an interesting topic as well to study the ethics of scientific research, because it's against a lot of things and it prevents and preserves life, even though we are trying to do research about such things. For example, you can't really, even doctors and scientists are studying the human body, the human anatomy. They do not just take living human beings, dissect them just for the sake of studying, right? That is unethical. What do they do rather? They take, maybe there are people who donate their organs, or corpses, people who've signed that when they die, they would like their bodies to be used for scientific research. At least there was consent provided there prior. So it's important to always get consent, especially if it's human being, because you cannot just do as you please. This is also a sensitive issue. So now, when you are done with that and you are sure that you have followed all ethical regulations and all hygiene factors, you wear the correct PPE. If it's your lab coat, your gloves, it's clean, you wear covered shoes, long pants. That is very important in a lab, because anything can go wrong, especially in chemistry labs where you're going to work with different chemicals. It can spill on you and burn you, but if you're wearing the correct PPE, then you'll be okay, because it will only touch on those external clothing you're wearing. Your glasses, your goggles, it helps. So be sure that you are following the correct procedures at all times. And then the stages in the dissection of a kidney. All mammals have two kidneys, right? There's the left kidney and the right kidney. A, when you dissect, measure or calculate the length and width of each kidney. Are they the same? So check that. B, you will remove the fat and describe the external features. Cut the kidney longitudinally. Longitudinally means you are cutting it over here, not like this. Wait, let me try and draw it for you. Longitudinally, this is the longitudinal, right? So how you'll be cutting, rather you'll be cutting this side there instead of this side. That is the wrong one, that side. And then you remove the fat. It's important, because when you find different kidneys, you'd find that there's excess fat surrounding it. The amount of fat will differ. Then C, look at the internal detail carefully and identify all of the regions and parts making up the kidney. Then you will draw what you see. Then D, use the stained dissection below and identify all the regions and parts on your own dissection. So this here is now stained. You stain it so that it can, the parts, the different parts can really show and can really appear even better now. Here's the video. Oh, I didn't, okay. The video we'll watch, the title is The Kidney Excretory System Dissection, GCSE A-Level Biology Need Practical Skills. So this is the title. If you wanna go look through this video on YouTube, type in this title. It's posted by Ava Hart's Biology. So go search for this on YouTube if you wanna watch it further after class. They really make great videos. I use them in most of my classes, the videos by GCSE. And they really help. They show you the practical of what we are doing. So all credits to these people over here. Here is that video. It will show you the kidney dissection, and then we'll discuss afterwards.
Hello and welcome to Samples for School Kidney Dissection. Today's video is about dissecting a lamb kidney, and when you receive your sample, it will come with the fat attached. This fatty layer is there to protect the delicate surface of the kidney organ. So the first thing we're going to do is remove the excess fat from the kidney. This should easily come away with just a few little nicks. So small little incisions around the edge of the kidney, and you can see the fat starts to want to peel away. We want to be particularly careful where the kidney joins on this side, because there's going to be vessels that we can investigate later. So very gently remove the fat without cutting into any of the organ. So once we've removed the fat, we can see the more familiar bean-shaped kidneys. Now I have a pair of kidneys here, so one would be on the left and one would be on the right of the body. And with our kidney this side, we can see the vessels that are attached. And there will be three vessels that you can identify at this stage: the ureter, the renal artery, and the renal vein, each of which come out from the same middle section of that bean-shaped kidney. When identifying the vessels entering and leaving the kidney, the ureter will be the vessel that is most covered in fat. The artery will be the thicker, and the vein being the thinner of the vessels. So on this kidney, I've trimmed off the excess ureter, artery, and vein to make cutting the dissection more straightforward. You can cut the kidney clean in half with scissors or scalpel to open it out like a book. Be careful not to cut into those vessels, and you simply snip all the way through. It's quite soft tissue. You can see you can start to open it. So the kidney organ is symmetrical, so you want to continue your cutting all the way down the center until you have two halves opened up. When our kidney is opened up, we can use a seeker to probe inside the areas that we can identify and then discuss the functions that they have. This outer layer of the kidney, just this darkish brown color, we refer to as being the cortex, identified by the blue pin. Moving inwards, each segment that you can see inside the kidney, we refer to as being a pyramid, identified by the pink pin. The darker red color, and a darker red because it's a higher density of blood vessels, is the medulla. The yellow pin. And feeding into the center area, we have what we call the pelvis. And this is the green pin. It's the pelvis then that feeds into the ureter that we cut off earlier in the dissection and transports the excretionary products out of the kidney towards the bladder. So when we are looking at the inner components of the kidney, the pelvis is a hollow structure because it feeds into the ureter. And extending from here are collecting ducts which go into the medulla. And if you prise slightly back the soft tissue of the medulla, you can see the tubules that connect. The collecting ducts reach into the medulla, which are connected to the nephrons. And there are approximately 1 million nephrons per kidney, and they are the functional unit of the kidney. So the kidney, of which all humans and mammals have a pair of, its function is to remove toxins from the body and regulate water content in the blood. This means that there is a complex process of excretion and reabsorption steps. As well as the kidney's function in osmoregulation, it also plays a part in blood pressure and calcium metabolism. For more information on how you can dissect a kidney and the role it plays in the body, and lesson plans and risk assessments, visit www.samplesforschools.co.uk.
Okay, guys, so that was the practical experiment for us. You saw the dissection of the kidney and everything we've shown you in diagrams. You can see it's real. You've seen it on a live kidney, right? Now, questions. You'll need to state a research question and a hypothesis for the investigation. What exactly, what is your research question and your hypothesis? Kidney is made of tubules called nephrons surrounded by blood vessels. That could be one of them, really. You can, there's, out of everything we've just learned and everything you know about kidneys, you can formulate any hypothesis that you can try and prove through that experiment. You can say that it's made of tubules called nephrons surrounded by blood vessels. Or you can say inside the kidney, there are structures called pyramids that you will find, or you will find a urethra. Really anything you can prove when you do the experiment. That could be your research question or hypothesis for the investigation. Remember, whatever hypothesis you choose, then it is the one that you'll focus on as you discuss. And in your conclusion, then you will state whether you accept or reject that hypothesis. Yeah? And then question 2, what precautions should be taken during the investigation? We explained a lot of the precautions beforehand. You need to use surgical gloves to avoid contact with blood and urine in the urethra. Then you also need to be careful with the scalpel or the sharp knife, because you are dealing with a kidney that contains waste material, right? That's where the waste material is processed. So wear gloves so that you don't get in contact with that blood or the urine. This applies not only for kidneys for human beings, this applies to all dissections where you'll be working with live tissue. It's very important that you remember this. For example, you'd never know what disease had infected that particular organism you're working with. Even if it's a human being who has passed on and you're working on their kidney, or even if they are still alive and you are doing some dissections in their body and you're trying to find something, maybe it's surgery for something, any condition, you wear gloves, because whatever sickness they have in their blood, you are most likely to get it. But if they have any type of infection, if you are not protecting yourself, then you are exposed to it and you yourself might get it. So these are necessary precautions, the gloves. And you also need to be very careful with a knife or scalpel, because they are sharp and they could cut you and then putting you more at risk, because there will be an exchange of fluids. If you happen to bleed whilst you have open alive, let's say you are, like I said, you are doing a dissection on someone who is alive, surgery, you cut yourself, then your fluids will get into their system. And what if it's you who has an infection and then now you pass it on to them? So it works in both ways. You need to protect both yourself and the person or the organism you're working with. In this kidney, however, yes, it is. It's on the outside of the body. You're dissecting it, so you need to protect yourself. Question 3: what do the cortex and the medulla look like? Guys, do you wanna indulge me and maybe give me your answers? What do the cortex and medulla look like? Do you know? Just seen it in the video. The cortex is that red-brown area about 1 centimeter thick. You saw that part there, that's brown on the edges. That is the cortex. It consists mainly of the microscopic section. It consists of mainly the macroscopic section, which is seen as the tiny dots. The Malpighian bodies, which are the renal corpuscles. And then the medulla is slightly lighter and is made up of the pyramids. So in that diagram, that cross section that we saw there, that very brown part outside or on the edges, it's the cortex. And then inside where it's lighter in color and you find the pyramids, then that is the medulla. And then next question is question 4. Identify and count the number of pyramids. This will differ, guys, because that video, it will differ based on when I counted in that video, I saw about 8 pyramids, but different kidneys, you have different number of pyramids, really. You will also depend on how big the kidney is. So in that video, there were 8, but maybe in yours, when you have a smaller kidney, you'd have maybe 6. Someone else might have about 10. So that will vary at all times. Question 5. What does the urethra look like compared to the renal vein and renal artery? I'm asking you now, this is posed to you. I'm going to give you like a minute or less to give me an answer. What does the urethra look like compared to the renal vein and renal artery? This lady in the video explained that. She explained how you can see or differentiate between the urethra, the renal artery, and the renal vein. So the floor is yours. You can type in the chat box or you can raise your hand so we can unmute you to talk to us. Okay, Butle is giving us an answer, kidneys to the bladder. So you're talking about the urethra here, ne? Okay, but how do you see when you're just holding the kidney and the bladder is not there, like this video? How do you see which one is the urethra? If it's a full structure, yes, and we are identifying a full structure, you can clearly see that the one connecting the kidney to the bladder is the urethra. But in such a case where we are dissecting the kidney and you can't really see where is the bladder, how would you know this is the urethra and this one is the renal artery and this one is the renal vein? Okay, guys, I'll provide that answer. The lady explained there that the urethra leaves the kidney and leads to the bladder, as Butle just told us as well. Ne? It is a little thicker than the veins and it is lighter in color. So the urethra is thick, very thick, and it's longer, right? Butle told us it's the one that connects to the bladder. Yes, Butle, yes, I got that. You are correct. It is thick though when you are dissecting and you see those 3, because it will be the urethra, it will be the vein, it will be the artery. The urethra will be thicker and lighter in color than the veins. And then the arteries would be shorter than the urethra, but thicker than the vein. So that's how you'd see the differences. The urethra is the longer one, and it would be thicker. It has more fat, and even the lady mentioned there's more fat surrounding the urethra than in the other 2 parts. Then the artery would be a little thicker than the veins, and then the veins are lighter than all of the other 2. Question 6. What are the dark, tiny spots in the cortex region? What are those dark, tiny spots in the cortex region? There are dark, tiny spots in the cortex region. The dark spots are what? The Malpighian bodies. Remember the Malpighian body consists of what? The Bowman's capsule and the glomerulus, right? Those ones are the dark spots. When we labeled and discussed the processes, I showed you the Bowman's capsule connected to all the other tubules. But now you can't really see them with you clearly if you are just looking at the whole kidney. But those very dark spots, the small ones, they play a very huge role in the processing and the filtration of the blood to ensure that it removes the clean blood from the waste product. So even though they might be that small, they are playing a very, very important role. And we discussed those processes yesterday. Okay, I showed you the answer. How do you safely get rid of the biological tissues and organs after dissection? So after you're done with dissecting the kidney or any other, maybe the liver, whatever will dissect in the future as well. What do you do with that biological tissue or organs after you're done with them? Do you throw them in a bin? Do you, what do you do? Where do you put them? The biological tissue is different. It's not like plastic or paper. You will burn it or incinerate it. Or what will you do? You will bury it. It's biological waste products. So that's what you do with it. You will burn it or incinerate or bury, because it's unlike any waste. Like we said, you need to wear your gloves to avoid contact with maybe the blood or the waste products. So now you can't, when you are done, you just throw it away. What about the people who actually sort out the bins? Maybe who look for waste, you can pass off many diseases and infections if you would just throw away biological waste. That's why you'll notice even in hospitals, there'd be bins specifically for biological waste, or let's say those components that have blood, and then there's bins for other types of waste. So it's always put in the correct place. You can't just throw away biological waste in bins. It would smell as well, it would stink, it would rot, it would cause more infections, more pathogens around. So it's not good to just throw away. So you need to be very careful with biological waste. And then now that we are done with the kidney, that part of the kidney, now you've seen the kidney, now you know the structure of the kidney, ne? You understand that? Are you okay? Are you good? We are now moving on to homeostatic control in the kidneys. Remember, we said kidneys play a huge role in ensuring there's a balance in water and salt concentrations. And then when we discussed the processes yesterday, you saw how it will be during ultrafiltration, the blood will be filtered, whereby the waste product will be taken on the capsular space, and then the blood will stay there in the vessels. And then after that, there'll be a process of reabsorption that happens. Remember, the reabsorption, you would either take in water or some salts if they are still needed in the blood, or even some hydrogen ions if they are needed in the blood. That's part of homeostatic control, to ensure the pH level is balanced, to ensure the water concentration is balanced, to ensure the salt concentration is balanced. And that balance that maintaining or that regulation process is called homeostatic control. So homeostatic control occurs in very, very many different organs across the body. The skin is responsible for thermoregulation, which is homeostatic control of temperature. And then the pancreas will be responsible for homeostatic control of glucose concentrations in your body. Medulla oblongata is responsible for homeostatic control of carbon dioxide. There are many different ones. Homeostatic control in the kidneys will look at how the water and salt balance is regulated. Unfortunately, yesterday we discussed a bit of the aldosterone and ADH. Now we'll see how they come into play. So at the end of this topic here of homeostatic control, you will know we will do the revision of the nephron, because it's important that you understand again the whole part and functioning of the nephron. Then you'll understand homeostatic control of the kidney in general. Then you look at it broken down, whereby it's controlling water and salts. The role of antidiuretic acid. Antidiuretic acid is the ADH. And then the role of the hormone, which is called aldosterone. So nephron, remember nephrons? You should know now. You should actually, I'm going to pose questions to you so you can answer me, because we've done the nephron. This is the functional unit of the kidney. Each bean-shaped kidney is composed of approximately 1 million nephrons. Each, do you understand this? Each bean-shaped kidney, one kidney, has about 1 million nephrons. That's why you can't really see them properly. There are many, many of them. The filtration unit of the nephron, which is the glomerulus, remember, when it filters the blood, this here regulates the concentration within the body of important substances such as potassium, calcium, and hydrogen. And it also removes substances not produced by the body, such as drugs and food additives. So you saw a bit of this. This is revision. The part of the nephron is revision for you guys. The filtrate, which is the urine, will then leave the nephron through a long tubule and collecting duct. Chemical signals triggered by the body's needs for water and salt cause the walls of the tubule to become more or less permeable to these substances, which are reabsorbed as required by the urine. So this part here, where the chemical signals involved and the reabsorption happens, it's process number 2, right? After filtration. Process 1 is filtration. Process 2 is reabsorption, whereby now it is these substances are reabsorbed from the capsular space through to the tubules here where this blood is, so that you ensure that the blood is regulated before it leaves the kidneys. So it needs to be clean and regulated. It can't go there and transport more drugs if there are drugs in the blood. It can't go and transport acidic blood if there was too much high loss of hydrogen ions. You understand? So this we discussed. This is the nephron here. If you, I can actually give you this. You can, if you want this, you can tell me and I'll supply it to you. Email me and I'll send it to you. If not, then you can. Okay, yeah. Actually, send me an email and I'll give it to you. This is some up a revision of the processes we studied in the nephron. The glomerulus filtrate, remember the glomerulus filtrate, that's where the process of filtration happens, ne? Useful substances are water, electrolytes, which are salts, amino acids and vitamins, and then waste would be urea, uric acid, glucose or creatinine. So what happens? Let's explain the structure again. The afferent arteriole, the structure and adaptation, this one has a wider lumen than the efferent arteriole. Remember, afferent is wide, efferent is narrow, and we discussed why. It helps the differences in these sizes. It helps with the blood pressure, so that more blood, the blood pressure increases, so that more blood is taken in quicker, and the filtration process happens more efficiently because of the differences in diameter. The other one is wide, the other one is low. So what do they do? To increase the pressure in the glomerulus to cause substances to be pushed out into the capsular space through the pores in the endothelial and the filtration silks between the podocytes. What is its function? To transport the blood from the interlobular arteries found between the renal pyramids into the glomerulus. So just a quick sum up there. The efferent arterioles, they are also involved in what? In collecting and reabsorbing the needed nutrients, which then become part of the vasa recta before it leaves the kidney through the venules and the renal vein. Then here, where this one, let me highlight it so you can see and follow through. The process there, proximal convoluted tubules, they are involved in the process of reabsorption, as well as the loop of henle. They play a huge role in the process of reabsorption. These two here, the arterioles, they play a big role in the process of ultrafiltration, as well as the efferent plays a role as well in the reabsorption, but the biggest one is in the ultrafiltration. So the proximal convoluted tubule, which is here, this one here, what does it do? The structure and adaptation of it. It is lined by simple cuboidal epithelium, which contains many mitochondria. This is important to know and remember. We discussed it yesterday with the short activity I gave you. Why the mitochondria? Because that's the site of cellular respiration, so there's a production of energy. Why do we need energy? for when the diffusion occurs through what? When the transport of these molecules occur in the process of reabsorption. One of the processes in which it occurs is active transport. The others are osmosis and diffusion, right? So they move through the process of osmosis or diffusion. But then the others also move through the process of active transport. Those ones are most likely to be the potassium ions and the sodium ions, which move through the process of active transport. And we know active transport requires an energy input. That's why these cuboidal epithelial cells have the mitochondria, so that there's the production of energy occurs on site. And then they have what? A broad, it's the microvilli. There's lots of microvilli there. What do? They increase surface area. Any organ with an increased surface area increases its functionality or its efficiency. That then assists in the reabsorption of the nutrients into the capillaries. So remember that. Now, guys. And then, what's the function of this proximal convoluted tubule? This one over here. The function is that glucose, amino acids, vitamin C, small proteins, and most of the sodium, chlorine, calcium, potassium ions are actively absorbed using ATP energy and a carrier molecule. So this here, when they say absorbed using ATP energy and carrier molecule, they are speaking about active transport. Most of the water is absorbed passively by osmosis. Osmosis is the transport of water. Diffusion is the transport or movement of substances. Remember the differences. And then we are discussing the ones that are involved in reabsorption, right? The loop of Henle, there. Loop of Henle is here. The proximal convoluted tubule is up there. The loop of Henle, its structure, it's like the ascending limb of the loop of Henle is lined with the squamous epithelium. The epithelium that is found in the proximal convoluted is called cuboidal epithelium. But the epithelium found in the loop of Henle is the squamous epithelium. The diameter of the tubule is thicker than the descending tubule over here. The ascending tubule, it has a thicker diameter than that of the descending tubule. And it is impermeable to water, meaning it does not allow water to pass through. It is impermeable to water. The descending limb, though, of the loop of Henle is lined with squamous epithelial as well, and that one is permeable to sodium and the chlorine, Cl ions, there's chlorine ions. See? So note those differences. What is the function? The tissue fluid in the medulla becomes hypertonic, and then water moves out by osmosis after the active transport of sodium ions, which is followed by the movement of chlorine ions. What do you mean by hypertonic? The tissue fluid is hypertonic outside, meaning it's concentrated, largely concentrated with these molecules outside. Then water will move out by osmosis. Remember, osmosis is the movement of water from a high water potential to a low water potential. So that's what happens over here. Then on to the distal convoluted tubule. Distal convoluted tubule, that one is also lined with cuboidal epithelium, the same way that the proximal convoluted tubule is lined with. This, which does not have large numbers of microvilli. So it doesn't have a large number of microvilli as compared to the proximal one. Then the permeability of the cells is increased with an increase in ADH secretion. There's that HDH that we are talking about, ADH. It's that hormone, right? The function is that tubular secretion occurs in both the proximal and distal convoluted tubules. Both sides, secretion happens there. And from the petubular capillaries, creatinine, hypuric acid, drugs, ammonium, potassium ions, hydrogen ions, and uric acid if the blood pH level is too low. Hydrogen ions are secreted into the nephron duct. This process is that of tubular secretion. Remember, you remember this word, guys. I hope you're not confused because we've discussed this and that's why I'm moving so fast around here. Then the very last one, because after it moves from the distal tubules, it will move all the way there until it's going out to the collecting tubules, collecting duct as well. Ne? This is lined with cuboidal to columnar epithelial cells, which are in single layers for easy reabsorption. Single layers again. So you need it to be thin in structure so that it can allow for ease of reabsorption. What is this function of this collecting tubule? The urine has been formed now. It's part of the urine formation. So what does it do? The urine will pass into this tubule from the nephrons. Water permeability is increased with increased ADH secretion, and the water is absorbed passively by osmosis into the hypertonic tissue fluid of the medulla. That is the process of urine formation. So you see how it all links up, right? And now it should make sense when we start discussing the homeostatic control. Guys, I didn't even notice the time. I'm so sorry. We are out of time. We'll continue this on Monday, innit? Before we start up with the revision for preparations for the paper. So yeah, I'm sorry, I didn't notice, but we are out of time now. Monday it is. We will finish up with this topic. Then we will start off with the revision, preparing for the assessment. Have a lovely weekend. Take care of yourselves. Wash your hands, wear your mask, practice social distancing. Bye.